Hello everyone. I'm Daniel Clarke, a data science analyst working under Dr. Avi Mayan on projects for Links, CFTE, and the IDG Knowledge Management Center at the Icahn School of Medicine at Mount Sinai. Today I'll be talking about Appyters, a software development toolkit we developed for turning Jupyter Notebooks into fully functional, standalone, web-based informatics applications. Appyters are both web apps and Jupyter Notebooks, hence the name. An Appyter consists of an input form, which allows you to upload data and/or configure aspects of the Appyter. Submitting the form triggers a workflow to produce a report. Some Appyters we've developed include customizable machine learning pipelines and omics data analysis workflows. We strive to produce detailed, transparent reports with publication-ready figures. The form shown here is from our most popular Appyter, accessible from Enricher. This Appyter visualizes enrichment results in the context of the underlying library. The Appyter Catalog is a public platform housing most of the Appyters we develop, making them searchable and operable on our cloud resources. About 100 Appyters are available today for performing a variety of bioinformatics analysis workflows and producing persistent reports. The next speaker, Sherry Shi, will highlight 3 Appyters and demonstrate their utility for hypothesis generation relevant to IDG targets, while I'll talk about Appyters more generally and from a developer perspective. Importantly, Appyters are extremely easy to produce based on existing informatics analyses that may already be implemented in the form of a Jupyter Notebook. Appyters provide a means to parameterize a Jupyter Notebook and use that notebook directly to construct an Appyter web app, turning one analysis into an analysis others can use even without coding experience. The functionality of the resulting application is strongly coupled with the meta notebook report, making the development lifecycle from prototype to production very short. For those of you who may be inclined to try out building your own Appyter, the easiest way to get started is by installing the Appyters off of PyPy, the Python Community Package Repository. The CLI contains a subcommand init for scaffolding a bare-bones Appyter project complete with demonstrative Appyter notebook and best practices for integration into the catalog if you choose to submit a pull request eventually. The README contains instructions on serving the Appyter from the Jupyter Notebook. The CLI also has many other functions which are documented on GitHub and in the CLI help menu. The way Appyters make a Jupyter Notebook into a parameterizable one is by permitting you to augment your notebook functionality with the help of Appyter Jupyter Magic. The first cell registers the Appyter functionality, and the Appyter code eval at the top here enables Appyter Jinja 2 metaprogramming functionality. This widely used template programming language permits variable substitution and flow control. Appyter provides a number of prepackaged fields, which are extracted automatically from your notebook. They are used to construct the form and substitute the user-provided input when it becomes available. Defaults are substituted automatically during development for quick testing, much like a standard Jupyter Notebook. The resulting app is fully functional, allowing tweaking of the parameters before rendering and executing the notebook on the fly in real time. The key idea behind Appyters is to derive everything primarily from a single source of truth, the Jupyter Notebook itself, letting Appyter developers focus more on the data science and less on the application development. The command line interface parses the notebook and builds the web application, and provides means to parse, construct, and execute Appyters outside of the web application context. It also includes helpful provenance features and tools for operating Appyters in production settings using distributed cloud resources. This contract allows the Appyter library to bring new features to Appyters that have already been developed. Some features in the works include OpenAPI and CWL specifications, and the ability to use custom WES endpoints for executing Appyters. In summary, Appyters enable informaticians to rapidly develop these web apps through minor modifications to existing reports. The apps are then trivially deployed and transparent to the end user who can access the actual code used for the report. We have used this framework to develop many different bioinformatics workflows and publish both the library and the catalog in Patterns in 2021. The Appyter Catalog can be accessed at appyters.minelab.cloud. I'd like to thank our current and past lab members, including summer undergrad fellows, most of whom have directly or indirectly developed Appyters and contributed to the paper. This work was made possible and is supported by the NIH through the CFD, Links, and IDG projects. Thank you for listening. I'm available to answer any questions you might have about Appyters at this time.